to the FDA. Okay, so we should understand the Ectd guidelines also. At least some basic Ectd guidelines we should understand, like how to organize the folders. So where to put the SDTM datasets, where to keep the ADAM datasets, where to add the patient profiles. So like that. So how to send the datasets to the FDA, then how to send your program to the FDA, then what type of document you have to send them, okay, arranging in a proper way. So, and we have different checklist also, okay. So before sending to the FDA, we have the checklist. So this is one example: study data standardization plan checklist, SDTM, ADAM, C dash. Okay, so this is about the checklist, general submission information. So anyway, we'll be discussing this. Okay, which version you are using. Okay, and we should write about the versions also. Then your define.xml. Okay, so like this. So this is a checklist before sending. So we should like check everything as per this checklist and send the content to the FDA. So this should be organized according to the ECTD guidelines, Electronic Common Technical Document Guidelines. Okay, so just a moment. Yeah, can you hear me? Avi, right? Yeah.
Yeah,
Okay, okay,
yeah,
fine.
I can hear you.
Fine, fine. Yeah, if you have any issue, you just ask me in between. Okay, then we'll be discussing about this ECTD also. Then about the statistic analysis plan. So this is very, very important one, because always we'll be dealing with the SAP only, statistic analysis plan interpretation. You should know each and every word in the SAP. So what is the per protocol? What is the study deviation? So what is the intent to treat? And how many types of study designs are there? So to understand the study, we should know the SAP. Then on which basis the objectives has been framed. Then what is the primary objective, secondary objective of the study? What is the primary endpoint? What is the secondary endpoint? Then what is the primary variable? What is the secondary variable? Okay, so what is your like efficacy endpoint? So these things you have to understand. So if you can understand the hypothesis, p-value, interpretation, then it will be easy for you to understand the SAP. And SAP contains different mock tables also, like lookup tables, mock-up tables will be there. So I'll be explaining them. Then trial designs also very important one. So we have different types of trial designs, like cross-control trials, randomized control trials, like parallel trials, unparalleled trials, then snapshot trials, like surveys. Okay, then factorial design. Like this, we have a lot of trials like single blind, double blind, multi-center trials, single center trials, like prospective trials, retrospective trials. Like this, we have different types of the trial designs will be there, and we will be discussing them in detail. Then MEDRA coding. So this MEDRA coding and WHO drug coding. So we have two types of the coding. MEDRA means it is the medical dictionary for regulatory activities. So this medical dictionary we use only for the regulatory activities, means mainly in the clinical trial designing and even the clinical like devices designing also, means to conduct any trial for the clinical purposes like medicines or antibiotics or any devices. So we use this meta coding only for reporting the adverse events, signs, symptoms. I mean, if any patient expresses any fever or any adverse event or any disease, so all those will be using the common terminology from the METRA, because people they use different terms. Some people they use the fever, pyrexia, hyperpyrexia, elevated body temperature, raised body temperature, like this. They use different terms. So instead of using those different terms, those words are called the verbatim words. So if you use the verbatim words, then I mean you will understand the fever. But in some countries they may not understand the fever. They may write the elevated body temperature, or they may write some up arrow showing temp, means the temperature increase like that. Or some people they will write the like 1 or 2 foreign degrees or 38 degrees centigrade like that. They won't write the word. They will just write the the numerical or metrics. So it will be difficult for you to understand if you are using different terms. So that's why we have to follow the MEDRA coding only. So I'll be explaining the Medra coding. I will install the Medra dictionary in your system, like trial version, so that you will understand. Because in the Medra we have different hierarchies, like low level term, high level term, system organ class, high level group term, preferred term. Like this, we have different, hierarchy will be there. So that hierarchical concepts you should understand. And how to do this Medra coding. So there's a lot about the Medra coding with the SAS. So with SAS we can do the coding, without SAS you can do the coding, and how many versions are there, which version you are using currently, and how to install it in your system. Like desktop version is there, and even SAS based versions are there, and macro based Medra dictionary is also available. Then who is maintaining the Medra? So these concepts I will show you practically. Then WHO drug. WHO drug means it's a World Health Organization drug. So it is about the drug. I mean, like uniformity to maintain the drug names. Like some people they use the Disprin. Some people they call it as aspirin. Some people they call it as acetyl salicylic acid. Means it is the same. The composition is same, but they use different terms. Even paracetamol, acetaminophen, both are same. But maybe they're using a different words, right? So instead of using those different terms, then better you have to follow the WHO drug names only. That is about the WHO drug. So, but mostly Medra coding and WHO drug will be done by the data managers only, not by the SAS programmers. But some SAS programmers who are from pharmacy background, okay, they will do the Medra coding, but not the WHO drug. So usually this WHO tag is maintained by data managers only. But Medra coding means those who are from pharmacy background, they are eligible to use this Medra coding. So non-pharmacy people or non-life sciences people, they are not eligible to do any type of the Medra coding. Okay, because according to the ICH guidelines, you should have proper knowledge and experience to perform a task in the clinical area, right? So that's why, say for example, if you have done pharmacy or any life sciences, then you can tell in the interview that you worked on the Medra. So if you are from engineering or from IT background, then if you say that I worked on Medra coding, then definitely they will catch you, because they won't allow you to do the Medra coding, though you are a SAS programmer, right? So there are some restrictions will be there who should use the Medra. Then coming to the domains. So these domains means you should at least remember some domains, like what are the HTTM domains, like AE, DM. So what are the variables present in the AE? What are the variables present in the DM? So those type of domains you should understand. At least some you should remember. And currently we are using the 3.2 version of the HDTM. So if it is a 3.2 version, then you should remember like how many classes are there, and in each class how many domains are there. Then totally how many domains are, just if you can make a notes like the domain numbers, okay, then it will be easy, and they will easily understand, or they will easily feel that already worked on the SDTM implementation. So you should make a list of the domains. Anyway, I will explain that. Then types of TLFs. TLFs means tables, listings, and the figures or graphs. So these tables, listings, and figures will be adding to the final study report. So what type of tables, listings, and figures usually we will be creating? Means basically we create only three types of the things. One is demographic tables. Second one is efficacy table. The third one is the safety tables. Always, what is ISSISC? Integrated summary of safety, integrated summary of efficacy. So this report contains the safety population and efficacy, sorry, safety report, efficacy reports, as well as the population report. Population is nothing but the demographic. So three parts we have. The SAS program has to keep three types of the reports in the final study report. Tables, listings, figures, and also demographic tables, then efficacy tables and the safety tables, right? So these are the three things we have to keep later. So anyway, I will be explaining you all this theory content along with the notes, and we have a lot of material also related to this. And I'll be providing some 6-7 books related to this drug development process, and I'll be giving a medical dictionary, the clinical trial dictionary also, so that you can use it for like to understand that particular terminology. For example, if you want to know what is an IRB, so you can just simply go through the dictionary, you will understand what is an IRB, what is an IEC, what's the data monitoring board, what's the data monitoring committee, okay, who is the investigator, what's the protocol, okay, what is the double blind, like that. So you can understand the entire terminology from the dictionary. So like this, I'll be providing some 7 to 8 books, okay, and also some books about this drug development process. How many phases are there? What is the first phase? What second phase? What happens in the first, second phase, third phase? Then what is our SAS programmer role in different phases? Then what is the post surveillance? So these things also, I mean, I will explain in that content. I mean, in those books you can find this content also. So it's all like about theory. Then coming to the second part, the programming. So in the programming, I divided this programming into 3 parts. One is just simply the basic things like validation, edit checking, importing the data and exporting the data using different procedures like SQL pass-through facility and accessing the data from Excel access like this. Because usually in the clinical industry, the data will be accrued or data will be collected and cleaned and locked by the data managers only. But might be, the SAS program also helps in data management area also sometimes, right? Because there is a data management area. If you got hide in that area, then they will be asking you to validate the data, then edit check the data. Even you are I mean, you are supposed to make the final clinical databases. Okay, so that time usually how the data will be collected in the form of CSV, or they will collect backend. Most of the electron data capture tools, what they do, they will collect the data at the, I mean, in the backend in the form of the MS Access or Excel or CSV files or Oracle. So you should know, if you have the data in Oracle, then you should know how to connect to the Oracle environment to import the data into your system using library engines or using SQL pass-through facility. So we have different methods to import the data into your SAS environment. So that we will be doing. Then you should know these simple procedures also, proxy port, simport. But usually proxy port and simport we won't be using in the clinical industry. So we use the library engines only for creation of the export transport files. But sometimes what they do by mistake, they will give you the export transport files by creating using the proxy port. So that time, if you are not familiar with the C port, then it will be difficult for you to retrieve them from the transport files. So that's why you should know the C port and import procedures also. Though we don't use these, if you use, then the FDA they won't accept because we have some guidelines how to create the export transport files. Okay, anyway, we'll be discussing those guidelines also. Then coming to the part two. So these are like simple thing: validation, edit checking, importing, exporting. So it's a simple thing. I think you know these in the basis also. Usually, what I do, I mean, somebody, I mean, who pursues the course from me, I used to teach these things in the basis itself. Okay. Then coming to the second part. Second part is a clinical SAS program, like common clinical program. So what's a common clinical program means here? You should understand how to define the variables. Means if you want to create a new variable, even in the SDTM dataset creation and ADAM dataset creation also, or while creating the tables, lists, figures also, we'll be defining new variables, right? So how to define a new variable means we have different methods like recoding and summarizing by using some advanced expressions. So we'll be doing, I mean, we'll be creating different types of variables like categorizing. So this is how to define the new variable. Anyway, we'll be discussing with the clear examples also. Then defining baseline observations. So we should understand how to define the baseline. Okay, so that we'll be discussing later. LOCF. LOCF means there are different methods to impute the content into the missing value. So those are called the imputation methods. So those imputation methods also I'll be explaining. There are different imputation methods are there, like how to work with the missing values. So for example, we have some data where some missing values are there. So you should decide whether to keep those missing values in your data or whether to remove those missing values from your data. How to decide means according to the SAP only you have to decide whether to keep the missing values in your, I mean, analysis part or not. So uh, for example, some analysis they won't accept the missing values. Okay, then do you need to remove those missing values, or you have to keep some values in the place of missing? Means you have to replace those missing values with some other value. Okay, when you are replacing, then which value you have to keep in that missing value? Means according to the SAP, then we use either LOCF. LOCF means it's a last observation carried forward, and that BOCF means it's a baseline observation carried forward, and WCF means it's a worst observation carried forward. Like this, we have different imputation methods out there. So there's a lot of content related to these imputation methods. We'll be discussing all these in detail later. Windowing the data. So what do you mean by windowing? How to pick up a particular value from the bunch of the results. See, for example, every day we are measuring the blood pressure for 30 days. Okay, then from those 30 days measurements, which one you have to pick, which one is the correct one? So that is like windowing. So how to pick the correct one from that bunch of the results. So that's called the windowing the environment or windowing the data. Then we'll be discussing this one also. Then transposing. So while doing the transposing, so I think you might be knowing this transposing. Even in the basis also you might have learned about the transposing and what are the statements we use in the transposing. So what the transposing do? Transposing is nothing but to change the dimension of the data set. Like some of the observations will be converted into variables and some variables will be converted into observations, and some variables they remain as it is. Like there are some variables may be dislocated. So these things will happen when you are doing the transposing. So what is the purpose of transposing? So without transposing, what type of analysis we can do? Then after transposing, then what type of analysis we can do? Then what common mistakes we do in the transposing? Even the SAS also it cannot identify those mistakes. So because when you're doing the syntax correctly, then the SAS will give the output. But the output may not be correct because you use the correct syntax only. Okay, but that is not the syntax error. It may be a logical error. So not only the syntax error, so there are some logical errors will be there. So those things usually we'll be discussing in the validation and the debugging thing. But you might be knowing all this in the basis also. In the basis, we have a topic, right? That is error checking or validation. So validation is we do the validation in different areas like quality control and quality assurance. QA and QC also will be doing, right? So what's the debugging? And what type of errors you will get: syntax error, data error, logical error, execution error, semantic errors. Like this we can find a lot of errors. So how to solve them? Data cleaning techniques also there. Okay, so there are separate books on the data cleaning techniques, like data cleaning techniques, debugging techniques. So we have a lot of content. So some I will just give you some data validation techniques later. Time to event. So time to event means what's the time to event? Time to sensor. How to calculate them? Okay. So these things also I will show you practically. Then change from baseline data set. So this is the main thing when you are creating the efficacy tables. Always efficacy tables means it's nothing but the change from baseline. So if you are creating some 50 or 60 efficacy tables means in all the efficacy tables, you can find the change from baseline concept. So how to write the programming for this change from baseline. Then critical variables datasets, categorizing the data, study day calculation. Okay, and not only this one, we have some other common programming techniques like how to use the proc compare, then how to do the meta coding. After meta coding, how to categorize it. So those common clinical programming things we'll be discussing. Then after this, the third part is implementation of the CDISC. So how to implement the CDISC standards in the HDTM, ADAM, and the defined XML. So everything I will show you practically this one. So these three are the programming part. Okay. Then the fourth one is after finishing this HDTM and ADAM, then we'll be working on the final study report creation. That is the CSR, clinical study report. In the clinical study report, mainly we'll be adding the TLFs, right? Tables, listings, and figures. So what type of tables we'll be adding? Demographic tables is the one type of table, then adverse event tables, then safety tables. So safety tables means always change from baseline only. So I will be showing you lab shift tables and some of the listings also. Because showing the coding for all the 100 tables or 200 tables is not possible, because everything they look similar. If you know how to create one listing, one table, and one lab shift table, that is more than enough. Okay, because all tables they look similar, but I will be giving a list of all the tables. I will give the data set, list of tables, and SAP specifications, everything I will provide. So if you are interested, then you use those SDTM datasets and convert them into the ADAM datasets, and from those ADAM datasets, you try to create a table using the SAS coding. Okay, so this is the the syllabus in brief. Then after this, what we do, I will help you to keep some projects in your resume. Then I will make you to crack the interview. Like if they ask any questions about your project or about the resume, then you should explain them in a proper way. Okay, so that I will show you. Then some interview questions, behavioral type of questions, that I will be showing, and also some, everything we will be doing, working on the real-time project, real-time only. It's not like dummy thing, right? So we have a lot of data, so which is already existing, and the data generated from the clinical trials only. So everything, it's a genuine only. So it's not like a created one or a dummy one. So it's all like live projects on. Live means like already finished and submitted, because the data is very confidential. So while the clinical trials are going on, before submitting to the FDA and after completion of the archive date only, the data will be generated. The data will come into the market, right? See, there's an archival period will be there. Archival period means like, okay, you are conducting a clinical trial. So after conducting the clinical trial, there is a repository will be there. A repository means the person like who is responsible to store the information. See, for example, there is a clinical research industries. There are some companies there, or some institutions, some organizations, some universities there. So in that university, the data repository will be there. Data repository means a person is responsible to store the entire information, whatever related to the clinical trial. He is responsible. Then after coming, the drug into the market, okay, so after 4 years or 5 years, then the archival period will be completed. Means the storage information will be, the storage period will be completed. Then that time you release the data into the market, no problem. So that time anyway, there is no use of the data. Even with your permission, the FDA also, it will publish the data into their websites. Okay. So whatever the data we are using now, so this is already like 6-7 years back data only. Okay, the 6-7 years back or 5-6 years back completed clinical trial data we are using, right? So yeah, so this is what I will do. And coming to the software, if you have 9.4 version or 9.3 version, then that will be good, because the older versions, like if you are using 9.1 version or 9.0 version, the macros, with the olden dates, then it's not possible to use the macros, because most of the macro variables we use, right? Like a table generated on system day. System day means it has to give the system day only, right? So if you change your system day, then it will give the old days. So while doing the calculations also, see for example, we want to calculate the age. So current date minus date of birth by 365. What's the current date? It's the current date is 2015. Then, but when you like change the system date like 2010, then it will give minus 5 years, right? So these type of issues will come, and some automation programs also will be doing in the advanced SAS. Okay, like automation programs means I want to generate some weekly report, monthly report, like bi-monthly report, fortnight report, like that. So I have to generate some periodical reports. So that time what we do, we won't be doing any type of the hard coding. So we'll be giving the system date itself. The system has to generate the reports according to the system date itself. So like scheduling, it will schedule. So when doing the scheduling also, we need to use the software with the current version. So this is one problem with the change of the date. And the second thing is the latest version, they support the proxy disk. That's the procedure, right? So if you are using 3.2 version of the SDTM implementation guide, let us assume, then if you have the 9.0 software, then in the 9. That time when the software released, like 9.0, so that time there was no the SDTM 3.2, maybe some SDTM 1.4 version might be there. so it won't support. So that's why we prefer to use the newer versions only. So if you have that one, no problem. Otherwise, I will send a link to you right away. So from that link, you need to download all the contents and keep ready in your system. In the next class, I will just do the installation, and I will make you to work with the current date only. So it will expire in 2016, not now. So you can use the same software for the next 1 year also. Okay. So yeah, so this is about, I mean, just the introduction. This is what I am going to explain in the clinical part. Okay. Then the second thing is uh, advanced, right? So we are talking about the advanced. So in the advanced, so this is I told you, right? Advanced will take 10 hours. Macros I'll be teaching for 3 days. Actually, here I didn't give the complete details. I will give you the even the topics also. Okay, I will send an email to you, or else I will give separately what topics I'm going to teach in day one, day two, and day three. The macros. So the macros will take 3 days. Everything like you should know the macro coding functions. Then after this, like I mean, like you will be able to use the macros and write the macros on your own, like how to store the macros and how to reuse the macros, auto call facility, and what is it, stored compiled macros. Okay, everything you will understand. Then 3 more days we'll be doing the SQL. So SQL means like single reports, multiple reports, joining the tables, and integrity constraints. Then, I mean, we will be comparing every topic. We will compare with the SAS SQL. So how to do the sorting in SAS and how to do the sorting in the SQL. Then how to add a new variable using SAS and how to add a new variable using SQL. How to make the queries using SAS and how to make the queries using SQL. Like this, I will be showing you the comparative program. So one is with SAS and one is with SQL. SAS, SQL, SAS, SQL, like this. Everything I will show with this SAS. And SQL, it's a structured query language. It is one of the oldest language. Okay. So the same language we use in the DB2 and the same language we use in the Oracle and MySQL, like in different databases we use the same language, but it may be slightly different, right? So origin of many languages, right, from Sanskrit only. So the Sanskrit was the old language. Okay, so most of the Indian languages are evolved from the Sanskrit only. Not only Indian, even some European languages also they evolved from the Sanskrit only. So the Sanskrit is having that much history. Just like that one, the SQL is a language which we use in different databases for making query. So the same SQL we can use in the SAS also. But the SQL language disadvantage is we can't make any analysis in the SQL, and we cannot generate any reports using the SQL language. It is just only for the data definition and the data manipulation. We can do the DDL and DML. That's it. So the SQL is the data definition language and the data manipulation language. So we can do only these two things, not the other analysis part and the reporting part, right? So anyway, it will take 3 days to understand the SQL, but no need to worry about the SQL. The macros is very important because as a SAS programmer, so what you do usually you will be using the SAS only for the coding. So we won't be using any SQL language for the coding, but just to understand. But some people what they do, they will use the SQL language only for their coding. So when you are not familiar with that, you can't understand, right? So nobody will ask you to write the SQL coding. Okay, you should write the coding and everything using the SAS only, but only while combining the tables. So we have some situations like Cartesian product or cross join. So how to do the cross join? So it's not possible with the SAS coding. So there you have to use the SQL only. So in that situation, then we have to use the SQL language. Then the SAS developed so many tools, like you might be knowing, like Enterprise Guide, Data Integration Studio, and SAS CDI, Clinical Data Integration Studio, SAS Standards Toolkit, SAS Clinical Standards Toolkit, and we have some SAS related tools. So many tools are there. So those tools, what they do, they automatically generate the coding in the SQL language. That is the main problem. So when you want to understand the coding, then you should know the SQL. So that's the reason we are learning the SQL. Okay, but no need to understand, like no need to have that much of efficiency to write the coding. If you can understand, that is enough. Okay, so anyway, 3 days the SQL will take. So 3 plus 3, 6 days to finish the macros and SQL. And 1 more hour we'll be spending on a project on macros. So I'll be giving you 1 or 2 projects hands-on, so that you will at least get some practice on the macros and SQL. So in 1 hour I'll be explaining a small project on the macros and SQL. So after this you will get some hands-on on both the SQL and macros. Later 2 days we will be learning the advanced programming techniques. Advanced programming techniques uh, index. What's an index? Then how to save the programming time, how to save the programming lines, like using arrays, something. Then benchmarking. Okay. Then what's the CPU time? How to reduce the CPU time? How to reduce the real time? Then there are some good programming practices will be there. Okay. Then those good programming practices. So those things, like if you have any doubts also, then those we'll be discussing in these 2 days. And coming to the certification, just certification guidance, like doing some dummy question papers, or like already existing sample papers. Okay, then sharing the material. If you have any doubt, then we'll be discussing on the daytime. Okay, so advanced SAS will take 10. So the clinical will take 15 hours, and I again in the clinical also I'll be giving you one before the first class. I will give you the complete like day wise what I'm going to explain the day one, day two, like that. I will give you the day wise syllabus. So here this is clear, right? And I will give the topics also. So it will take 10 hours and the clinical will take 15 hours. Totally it will be 25 hours. Okay. Yeah, so this is about the course details. Okay. Then yeah, it will be 1 hour per day. Then during the weekends also, if you're interested, then I will spend more time to finish it. Okay, then yeah, I will show you some important websites. Just go through them. One is the ICH. ICH means it's an International Council for Harmonization of Technical Requirements for Pharmaceuticals for Human Use. So International Council for Harmonization. So actually, any pharma company who are manufacturing the drugs for human usage, they have to meet all the technical requirements defined by the ICH. ICH is a international council. So there are so many members are there in the ICH, but the ICH started with 3 countries: Japan, European Union, and the US. So these 3 countries, they combinedly and collaboratively, they developed these guidelines called the ICH. Now so many countries were added in the ICH. Okay. So we divide the world into 2 types of the countries: drug manufacturing countries and the non-drug manufacturing countries. Like if you take some African countries, they won't manufacture any drug. So drug manufacturing countries are less and non-drug manufacturing countries are more. Okay, I will give those statistics also. Okay, then but these drug manufacturing countries are providing medicines to the non-drug manufacturing countries. So the US is a bigger one, like which is manufacturing the drug. Okay, then even India also, it's in a good place, like manufacturing the drugs. So like what is the proportion? Okay, India and US and even like UK, and what is the China role in preparation of these drugs? So what type of drugs they are creating? So those concepts also will be understanding. But in the ICH, who developed this ICH? So this history also will be discussing. But if you can go through this site, you will understand at least something, some concept you will get. In the ICH, there are different types of the guidelines out there: QSEM, quality, safety, efficacy, and multidisciplinary. So we have some quality guidelines. Quality means like usually the SAS programmer need not require all these quality guidelines, because we never involved in manufacture of the drug, right? So we never bother about the quality of the drug. Those who are preparing the drug, they need to follow these quality guidelines, like good manufacturing practices. Then the drug should not contain any impurities. The drug should be stable. It should not evaporate. Then it should not be exposed to light. If it is exposed, then the content of the potency of the drug may be reduced. So what to do? Like not to like lose the potency of the drug, like this. So these are all the quality guidelines used by the manufacturers, not by the SAS programmer. So no need to worry about this one. Then coming to the safety. Safety also, the drug is carcinogenic or the drug is toxic or the drug may cause some other adverse events. So we never bother about all those things. Only the people who manufacture the drugs. So they will bother, means mainly the R&D department. They will bother about the safety things, right? Then the third one is efficacy. So here we play an important role in the efficacy, because how to find out the efficacy? The efficacy will be, I mean, diagnosed or detected by conducting the clinical trials only. So as a SAS programmer, we will be handling the clinical study reports, right? So here you should know the three things. One is clinical study report, because we play an important role in creating the clinical study report, CSR. So this you should understand. What is the structure and content of the clinical study report? I will just brief you. Then E6, good clinical practice. Everyone should know this GCH. I mean ICH GCP guidelines. So good clinical practice. Then the third one is the general considerations for clinical trial. So what is a clinical trial? This is a general consideration, like who is the investigator, who is the data manager, who is the investigator, then what is the role of the pharmacist, what is drug accountability. So these things, the general considerations for clinical trials. So we should know these three. If you are more interested into the biostatistics side, or if you're from a statistical background, or if you're interested into more analytical side, then you can go through this E9 also. E9 means it's a statistical principles for clinical trials. Mainly this is for the statisticians, not for us. Those who create the SAP, then they have to follow these statistical principles. But if you are interested, even you can go through this one. So these three we needed, E3, E6 and E8. Okay. Then the fourth one is the multidisciplinary. Multidisciplinary, all the other things comes from the multidisciplinary. Here in the multidisciplinary, we'll be learning these two things, electronic common technical document and common technical document. So how to send electronically will be explained by ECTD. So how to send manually will be explained by the common technical document. But currently we are using the ECTD only. If you just understand the concept, that is enough, because we never involve in sending the reports or anything to the FDA, right? So there is a separate regulatory team will be there, regulatory affairs team will be there. Like in US also, we have some 2 years courses on the regulatory affairs, right? So 1 year or 2 years courses are there in the regulatory affairs. So those who are pursue those courses are the subject expertises. They will send the ECTD things. Okay, so you need not to bother about this. Then meta terminology. So we'll be learning this meta terminology and hierarchy also. Okay, so just go through this website and browse and try to download these things also, and just have a look at this one. This is an ICH. Later, you should know the Nuremberg clinical trials, right? So what is the Nuremberg clinical trials? During the Second World War, actually what happened, the soldiers, uh, soldiers got captured, see, held for the purpose of bringing Nazi or criminals to, okay. So here they conducted some clinical trials, okay, series of 13 trials carried in a place called the Nuremberg, Germany. Means they captured the soldiers as a war criminals, and they conducted the clinical trials brutally on those people. Okay, means considering them as a animals, laboratory animals, they conducted so many clinical trials. So it's a non-ethical, right? So that time, World Medical Association, they met, okay, in a place called the, uh, I mean, it's Finland, right? So Finland. So and they proposed some like ethical guidelines. Okay, that's called the World Medical Association. Okay, Declaration of Helsinki. So they declared some guidelines in the place Finland. The place is called Helsinki. So in the Helsinki, see, WMA, Declaration of Helsinki, ethical principles for medical research involving human subjects. When 18th WMA in 1964, but they met first time in the place called the Helsinki, Finland. Okay. So after the Second World War, after this Nuremberg clinical trial, so from these only the ICH guidelines got evolved. Okay. So you should download this PDF. Okay. Download the PDF and just have a look at this WMA. I mean, ethical principles also. Okay. This is also very important thing. So yeah, this is about this WMA Declaration of Helsinki. This website, and go through this website also, medra.org. It is about the medical dictionary for regulatory activities. So this is maintaining by a company called MSSU, Medra, I mean supporting organization, software supporting organization. So it is about the Medra. But anyway, I will be explaining like which version you are using, how it looks like, what's the hierarchy. Okay. Then we have one more website, that is clinicaltrials.gov. Clinicaltrials.gov. Yeah, gov. So what the clinicaltrials.gov do? It is a clinical trial registry. Registry means if you want to find out any clinical trial, you just search here. For example, if you want to know like diabetic clinical trials or any clinical trial, you just type here and search. Then it is showing 13,220 studies for diabetes. Some are completed, some are recruiting, and some are terminated, some are unknown, like this. But most of the diabetes trials and what is that psychiatric trials and what is the other one, obesity related clinical trials and cardiac disorders, heart attack, like that. So I mean, because of the lifestyle, right? So all these are coming. Cancer, mainly cancer. So these type of clinical trials you can find mostly in US only, because I think the US is the #1 place where you can find more cancer patients. See, 51,433. I mean, we search for the diabetes, showing some around some 20 or 30,000, but here the cancer trials are 51. You take any cancer trial, so you can find in US only, because I think the US is the first one in, oncology trials. So nowadays, but the one thing here, as a fresher, you should not show anything related to the oncology therapeutic area, because uh, the normal, all the clinical trials report will be different from the oncology clinical trials. Okay, if you write any word about the oncology in your resume, then definitely they will ask you about the oncology and you will get placed immediately also, because the oncology is a bit difficult area in the clinical trial, and even the oncology tables, even the oncology the reports, everything look bit different from our normal clinical reports, and you should know some terminology also related to the oncology clinical trials. So that's why they will ask, and they will search for the oncology expertise also. Okay, but after working genuinely for 1 or 2 years in the clinical industry, then you can go for the oncology clinical trials. Okay, so yeah, but you can find a lot of clinical trials in oncology. Okay, so I'll be providing you some information. Like I have 1 or 2 books related to the oncology clinical trials. If you can go through and try to understand the terminology, then definitely that will help you in future. So you can search like country wise also. So it's a registry. Okay. In India, 2,952. Very few. Very few. Okay. So United States. See, 91,000. Okay. You can't compare it. 2,000 and it's almost 1 lakh. So it's about the registry. The registry also, they didn't maintain, the trials. See, for example, some clinical trial they started 16 years back also. Still, they are doing the trials only. then those information you cannot find here. Means I think they are maintaining this from last 6 7 years. Okay, 6 7 years trials only, but not the complete one. Okay. Okay, so this is one more website, and you can find the clinical trials completed from different websites also. Like you take the Novartis, okay, you can find what they are conducting, and you can access their protocols also. You can access their SAP also. So you connect to Rocky and PPD, and so many organizations, Klein became SB. You just connect, browse their website, and you browse the US FDA website also, where you can find download a lot many data sets and the protocols, SAPs, even the ADAM and HDM data sets also you can download. Okay, so you can just browse those websites also. Then we have one more, that is cds.org. So this cds.org is a clinical data interchange standard consortium. It's all about the clinical data only. It's not like how to conduct the trial. It's not like how to, recruit the patient, or how to make the progress in the clinical trial. And it is not like submissions. So it's all about the how to manage the clinical data, how to collect the clinical data, how to validate the clinical data, how to generate the reports, how to organize. So it's all about the clinical data standards only. So this will help us to maintain the uniformity in the data, right? So multiple companies, if they follow their own standards, their own guidelines, then it will be difficult for the FDA to cross check all those things. So that's why the main concept of the CDISC is to maintain the uniformity in the data, right from protocol creation to until sending the until submitting the reports to the FDA. In each and every level, we have to follow the guidelines. So those guidelines are mentioned in this CDISC. Even the CDISC also will be led by the regulatory submission people only, or some people will be hired separately, those who work on the CDISC. But as a SAS programmer, you should know the SDTM, ADAM, and the define XML. So these three things I will be explaining. Okay. So these three things I will explain with project also. Okay. So anyway, I will make you very perfect in these three things. Okay. But we have different methods, but I will be using the SAS only. Okay. So just go through the CDs and download some, what is the mission, and what is the strategy and goals, and what type of standards we have. And you can find some videos also here. Even in the YouTube also, you can just find some videos on the CDs, but a bit difficult to understand. Okay. If you know the concept, then you will understand. Okay. So it is a bit difficult to understand. Okay, then after the training, then if you can listen, then definitely you'll at least get the concept. Okay. Yeah, so this is our course overview. Okay. So yeah, let me know your interest through an email. So if you want to continue, then no problem. You just trust me. I will continue the classes, because if I have only one student, then might be it's not possible. But I mean, you both are attending, right? So that I will continue. And till Friday, like tomorrow, yeah, tomorrow I will start the clinical part. Then Friday, we will shift the clinical class to some other time, and Friday onwards we'll start the advanced. I will start the macros. Okay. So simultaneously, 1 hour we'll do the advanced, and 1 hour we'll do the clinical. Okay. So that we can finish everything in 15 days. Like by the end of this month, we can finish the clinical as well as the advanced. Okay. If we do the continued classes, and if you spend more time during the weekends also, so you'll get the perfectness till, I mean, the end of this month. So we'll make a plan. Okay. So the days and the content. So based on that, we'll work on. Okay. Yeah. That's it. Okay. So